Okay, ladies and gentlemen, welcome to our biology lecture class. In this lecture class, we are going to discuss lipids. Specifically, this is our part 2 of our class. So let's quickly pick it up from where we ended, and let's start by having a review on fat acids. So if you look at fat acids, like we said, fat acids are made up of two components. They are made up of the head, which is made up of the carboxyl group, and it also made up of the tail, which is made up of the hydrocarbon chain. So fat acid is just made up of a hydrocarbon chain, which I've already stated, and this hydrocarbon chain, it is the one which is often or which is hydrophobic. That simply means that it doesn't interact with water. Since the fat acid has the polar head and the non-polar head, that simply means that the fat acid is amphipathic. So this means that it is able to interact with water and also not able to interact with water at the same time. For more explanation, you can kindly check out our class part 1 where I explained about that. So classification of fat acids. So this is where I'll now go a bit in detail and explain. So fat acids are classified into two groups. So there are two groups of fat acids. This depends on the presence or absence of a double bond. So fat acid can be classified as saturated fat acid and also unsaturated fat acid. So we have two types of fat acid: saturated fat acid and the unsaturated fat acid. Like in this diagram, if you check, there is the saturated fat acid, it doesn't have the double bond, and the unsaturated fat acid, it normally has at least one single double bond. So this is one of the major differences between the saturated fat acid and the unsaturated fat acid. So let's quickly look at what saturated fat acids are and what are you supposed to know. So these are just fat acids that do not have a double bond. So when you talk of saturated fat acids, they do not have a double bond, and this simply means that they have a maximum number of hydrogen atoms. So these type of acids, they normally have a maximum number of hydrogen atoms because they do not have a double bond. So they have no double bond. The other thing that you need to understand is that they only have a carbon to carbon single bond. So you can only see single bonds between the carbon atoms. Then the absence of these double bonds makes saturated fat acid rigid, and this is one thing that makes these saturated fat acid to be what? To be solid at room temperature. So one thing we are going to realize is that because of these guys, they don't have the double bond, they tend to have a rigid structure, or the particles are closely packed. So the particles, or you can call them molecules or whatever you can call them, they are normally what? They are closely packed. So they are closely packed. So that's one of the part you need to understand. So this rigidness makes fat acids like wax to be what? To be solid. So fat acids are normally solid at room temperature. The other aspect you need to talk about saturated fat acids. These fat acids have a linear structure. They have a linear structure. That's one part you need to know. Then an example of a fat acid which you will be asked in the exam is the stearic acid, and this stearic acid has just the 18 carbons, and the chemical formula is simply this one. So please make sure that you are able to know the chemical formula of stearic acid, and you are also able to know the number of carbons which stearic acid has. So apart from what is on top, just on this part of example, there are about 3 questions you can be asked on. One can be referring stearic acid as an example of a saturated fat acid, and also the number of carbons it has, and you can also consider the chemical formula of stearic acid. The stearic acid is found in things like butter. So most of you have eaten butter before. In cheese, I know majority of you have never tasted cheese. And then coconut oil, some of you have not even seen it. Meat, everyone has eaten meat before. Margarine, you can go in supermarket, you shall see it. And then fried food, most of the fried food, they have this acid which is called what? The stearic acid. So stearic acid in this case is just playing a role as an example of a saturated fat acid. So it is mainly used in the production of detergent. So the detergent, you have the detergent paste, you have many detergents which you normally use to wash your dishes, to wash your clothes. All those detergents, most of them, they are made from what? From stearic acid. Even things like soap, shampoos, and also the shaving creams. So for our colleagues who have reached puberty, I'm sure you are familiar of shaving creams. Yeah, we have shaving sticks and shaving creams. So all those guys, they just come from what? Stearic acid, which is an example of a saturated fat acid. Okay, with this information, we can look at this picture, which is just showing an example of a what? A saturated fat acid. An example of a saturated fat acid. If you look at this picture, there is no double bond. Okay, there is no double bond. So since there is no double bond, this guy is a what? Is a saturated fatty acid. Okay, this is the structure of stearic acids. So please make sure that you know the structure of stearic acid. Please make sure that you know how to draw, and also how to identify the structure of stearic acid. So stearic acid, like I have said, is just an example of a what? Of a saturated fat acid. So make sure that you put that in mind, and make sure that you know how to identify it. So let's quickly look at some things that are made from saturated fat acid, especially stearic acid. So in this case, you look at butter, which most of you know. So butter, it normally comes from a saturated fat acid. So most animal fats, if you look at most animal fats, they are solids in nature, and they are examples of saturated fat acid. And they are solid at room temperature, like I've already stated. These ones, make sure that you limit them in your diet. The reason why you are limiting them in your diet is that they can contribute to conditions such as heart disease. Especially in the current years, you find that a lot of people are experiencing heart disease and many cardiovascular diseases. This is due to the intake of much of the saturated fat acid. And you know that these guys, they will automatically whether you boil them or maybe you fried them, they'll automatically go and stick in your what, in your blood vessels, and they will normally go and cover up your heart. As a result, you find that you might find yourself having cardiovascular diseases, or an example, it can be a heart disease. So ladies and gentlemen, know what you eat and make sure that you take care of your health. Like I've already stated, they can make a solid in the arteries, the blood vessels. I'm sure most of you understand the blood vessels. Let's quickly look at the unsaturated fat acids. The unsaturated fat acid. So fat acid with one or more carbon double bonds are said to be unsaturated fat acid. This simply means that they have less number of hydrogen atoms. And then monounsaturated fat acid have one carbon to carbon double bond, while polyunsaturated fat acid have more than one double bond. Now, the presence of a double bond creates a bend. I think in part 1 I explained that a bend is also known as a kink structure, and this makes them to be flexible and able to melt at much lower temperature than the saturated fat acid. So with this reason given, you find that these guys are normally liquids at room temperature. So the unsaturated fat acid, they are normally liquids at room temperature. Oleic acid is an example of a monounsaturated fat acid with 18 carbons, and this is the chemical formula. So please make sure that you know that oleic acid is just an example of the unsaturated fat acid, and it only has a single double bond. It only has a single double bond. So please make sure that you take note of that. Then this guy is oily liquid. Like I've already stated, most of the unsaturated fat acid, they are liquids at room temperature, and they are normally you can find it in olive oil, you can find it in avocado, and you can find it in soya beans, you can find it in canola oil, you can talk about sunflower oil, and also walnuts. So if you look at the unsaturated fat acid, these are healthy fat acids, these are healthy food. Okay, most of them, like let's say avocado, it has a lot of healthy benefits. You can talk about soya beans, of which it has a lot of health benefits. Even sunflower oil, it has a lot of benefits as compared to the previous ones which I've already mentioned. So this is how the structure of unsaturated fat acid, of which the example is oleic acid. And you can see there is a bend there which is also known as a kink. There is a bend there which is known as the kink. So this is the reason why these guys are normally liquid at room temperature. One thing you need to understand, that unsaturated fat acids are not linear. They are not linear as compared to the saturated. The saturated are linear, but the unsaturated are not linear. They have a bend there. Then the same unsaturated fat acids, because of the I'll allow some questions after I explain this part. The same unsaturated fat acid, because of the presence of the double bond, the unsaturated fat acid can be classified into a cis and the trans. You're going to understand more about cis and trans isomers when you do your organic chemistry with your organic tutor. So cis unsaturated fat acid have hydrogen bond on the same side of a double bond, making them relatively more flexible than trans fat acid, which have hydrogen atom on opposite of the double bond. So in biology, the most important thing about the cis and trans, you have to know that for the cis, the hydrogen bonds are on the same side and then they are more flexible. So this is a very important point that you need to take note. Then for the trans, for these guys, normally they are not much flexible because the hydrogen bonds are on the opposite sides. So that's how a cis fat acid, if you can see, the hydrogen bonds are on the same side and this guy is more flexible. This guy is more flexible. Then for the trans, the hydrogen are on opposite side. For the trans, the hydrogen are on the opposite side and this makes it to be less flexible. Okay, at this moment I'll allow you guys to ask questions. Before I move on and look at an alcohol called glycerol and then go further and look at phospholipids, triglycerides, waxes, then I'll call it a day. So we are free to ask questions at this moment.
Regarding the formulas for saturated and unsaturated, I have seen a lot of questions which says state formula for unsaturated. Because if you say that unsaturated, the formula for steric acid has 18 carbons, does that mean that all the saturated waxes have the same formula like steric acid?
You mean the words for saturated?
Yes, saturated. They ask a question, you choose the formula for the saturated fatty acid. Is it the same one? Am I supposed to choose the same one for steric acid? Is it the only fixed one like that?
No. You need to follow that formula. Please mute yourself. You need to follow this formula here. This is the formula you need to follow for the saturated fat acid. So this is normally the basic formula you're going to follow. So X can be any number. So that's for the saturated fat acid. But for the unsaturated fat acid, these guys, they normally differ. Okay, for the unsaturated fat acid, these normally differ. So they have a lot of formulas. Depending on the number of bonds that they have, depending on the number of double bond which they are having. So if it is having one double bond, then remove 2 hydrogen. Okay, if it is having one double bond, remove 2 hydrogens. If it is having 2 double bonds, remove 3-4 hydrogens. I hope people are getting me right there. I'm saying for the saturated, this is the fixed formula for the saturated. Okay, this is the fixed formula for the saturated fatty acids. Now for the unsaturated fat acids, you need to make sure that you remove 2 hydrogens in a case you have a single double bond. So if you have a single double bond, remove 2 hydrogen, okay? And then if you have 2 double bond, remove 4 hydrogens. Then if you have 3, remove 6 hydrogens. So that's how it goes. And sometimes it depends on the place where the double bond has been formed, but in most cases, when it is 2 double bonds, when like this guy here, since it has 1 double bond, so you remove 2 hydrogen, okay? Then in a case you have one which has, let's say, which has 2 double bonds, then you remove 4 hydrogen atoms from it. So that's how the game goes. Okay, I think with that being said, I hope you guys have gotten 1 or 2 points on that. The girl who asked, is it okay on your side for the formula?
Yes. Yes.
All right, thank you. So for the formula, a lot of questions they come on the formula issue. They come, a lot of them. So try to find out a formula for, let's say, 1 double bond, 2 double bond, 3 double bond, 4 double bond. You can go up to 5 double bonds, and then you can rest there. Okay, let's move on and look at Grisello. Let's move on and look at Grisello. So Grisello is a common alcohol. So Grisello is a simple 3-carbon polyalcohol which combine with fat acids in the synthesis of glyceride lipids. So Grisello is just an alcohol which plays a role in the formation of glyceride lipids. Then it is colorless in nature. It is colorless in nature. It is viscous and it tasty sweet. So that's the formula. That's the property of Grisello, which is just an alcohol. So the 3 hydroxyl group make it water soluble, and it's able to interact with the water due to it having that the hydroxyl groups. So these are the hydroxyl groups. So these hydroxyl groups, they are the ones that makes this glycerol to be able to dissolve in water. And the chemical formula for glycerol is C3, okay, coming from 3 carbons, and you should make sure that you know how to draw it. And then H5, and also you can say OH3. The oxygens are 3, which is the 3 outside, H 1 2 3, and then there are 1 2 3 4 5 5 hydrogen. So this is the formula for Grisello, or sometimes it can be written as CH2 and then OH. You have that, then you have CHOH, and then you have something like CH2OH. So these formulas are very important in your tests and exams. So I've already written that in red ink. So that's just the chemical formula of Grisello. So this is the chemical formula of Grisello. You'll find it in the final exam or test. And this guy is normally produced from the lipid through saponification, which is just the reaction between sodium and a glyceride. So a glyceride is just a combination of a fat acid and the glycerol, which is just a lipid. And often it is used as a solvent, a sweetener, a preservative, or as an additive in food. And for a friend who make pharmaceutical and cosmetic product, they also use this same glycerol. So that's just another way of representing glycerol. So that's another picture which is simply showing glycerol. With this being said, I'll go further and look at glycerides. So for now, do you guys have any questions on this particular part? Any question? Okay, so that means that we don't have questions. So let's move on and look at the next part. Glycerides. So glycerides are just esters or compound of glycerol and the fat acid, of which you know that glycerol is an alcohol and then fat acids are just what? Acids. So when these two combine, they are forming what we are calling a what? A lipids. And this is in line with the definition which we had in part 1 of this lecture class. And then each of the three hydroxyl group is able to react with the carboxyl group of a fat acid. At this point, I'm interested in you knowing who is reacting with who. So the glycerol, it contributes the COH, is the one which is reacting there. And then the fat acid, it is utilizing the carboxyl group. So the carboxyl group is reacting with the hydroxyl group. So the carboxyl group is found on the head of the fat acid, and then the hydroxyl group is found on the glycerol, which is an organic alcohol. And then the bond which is formed between the hydroxyl group of glycerol and the carboxyl group of fat acid is known as the ester bond. And this is again is in line with the definition which we made. And remember that we said that during the formation of this bond, there is the production of water. As a result, this is a condensation reaction. So this is a condensation reaction because water is normally released in the process. Then when you look at this same guys, you have a monoglyceride which has only one fat acid chain, meaning only one water produced, so one H2O produced. Then there a diglyceride has 2, so this one has 2 water produced, 2 H2O produced. Then the other one, it has 3, which is the most common one, and this one has 3 H2O that have been produced. Okay, so this is just the formation of triglyceride from 3 fat acids. So if you look at this point, you need to understand that the fat acid is using the carboxyl group to react with the hydroxyl group of the glycerol. Another exam question which comes is who contributes OH and who contributes H. So in this case, the guy who is contributing OH, it is the hydroxyl group of of glycerol, and the one which contributes H, it is the what? It is the carboxyl group of the fat acid. So when these react, they'll produce the water, which you can see there. When these also react, they'll produce water. So this reaction is a condensation reaction. Now, in case you want to take the triglycerides to these two guys, then you have to add water, and this reaction is called what? Hydrolysis. So hydrolysis is just the reverse of a condensation reaction. Okay, let's quickly look at phospholipids, and then I'll look at wax, and I'll look at steroid, then I'll allow you guys to ask questions. So for phospholipids, one thing you need to look at, it has a phosphate group. It has a phosphate group. It also has the glycerol there. So this phosphate group, the glycerol, they make up the hydrophilic head of a what? Hydrophilic head of the phospholipid. Then automatically the fat acid, it is contributing to the what? To the formation of the hydrophobic tail. Then one thing that you need to understand is that in phospholipids there are two types of fat acid. So normally it will exist as saturated fat acid and the unsaturated fat acid. No phospholipid has only saturated fat acid or only unsaturated fat acid, because these phospholipids, they play a lot of roles in the cell membrane. So the long hydrocarbon chain of the fat acid form hydrophobic tail, which makes lipid insoluble in water. I think this was explained in part 1 of our lecture class. Triglycerides are the most commonest lipids in nature. Please do not mistake this guy to phospholipids. Phospholipids are most abundant in a cell, but triglycerides are most abundant in the what? In nature. And then these triglycerides can be classified as fats and also as what? Oil. So mostly they are solid at room temperature, and these ones are liquid at room temperature. So the same triglyceride guys, they are of two types. You have the fats, which are solid, and you have the what? The liquids, which are what? Oil. So fats, it is solid, oil, it is liquid, but they are what? They are triglyceride. By the diagram here, it is talking about the phospholipid, which I think I'll talk about it later on. Yeah, that's the phospholipid. So this diagram here, it is for phospholipid, but this explanation, it is just an added explanation on triglycerides, which are classified into fats and the oil. I think I've already talked about phospholipids. Like these phospholipids, they are able to form a bilayer sheath in the cell membrane. There is a bilayer sheath. So this guy, it plays a role in the permeability of some substances through the cell membrane. Then they are also able to form the micelle. So we have a word which is the micelle. This is whereby now they are not able to dissolve in water, but they will form a cluster-like structure, and this cluster-like structure which they form in water is called a micelle. So this is a property of lipids. This is a property of lipid which is unique to them. Okay, and then if you look at liposomes, these guys are normally spherical lipids, and they are usually 50 to 500 in diameter particle size, and they have one or more lipid bilayers. So these guys, they more like appear like the cell membrane. So phospholipid is just a modified triglyceride. That's the reason why I had put a phospholipid on the triglyceride. So phospholipids are just modified triglycerides in which one fatty acid has been replaced by a phosphate group linked to a nitrogen containing molecule, like this one. So if you look at a phospholipid, it has the saturated and the unsaturated. Now for the triglyceride, here you expected a fat acid, but in a phosphate, that's why it is called a modified triglyceride, in such a way that the fat acid which was supposed to be here, it is being replaced by a phosphate group, and this R group, it is just a what? The R group, it is just the nitrogen containing molecule. Now one of the two fat acid chain of phospholipid is saturated, while the other chain is unsaturated and has a kink structure. So this is very important for you guys. Now phospholipids form a ball-shaped micelle or liposome in water. I think this has already been explained when I was talking about these diagrams. So this is because of the hydrophobic interaction between the fat acid chain, which tend to cluster inward away from the water, while the phosphate group stick out into the surrounding water. So in some exam questions they will use a micelle, and in other exam questions they normally use a liposome, so be careful. So that's just a structure of phospholipid which is being found in the what, in the bilayer. Remember, if this side there is water, so that's where the head will face, but the tail will normally face outward. So you have a saturated fat acid, unsaturated, saturated, unsaturated, just like that. Then inside the head you find the phosphate group, which is attached to the nitrogen compound. Okay, let's quickly look at waxes. So if you look at the diagram there, it is just showing an example of a wax. So a wax are mainly esters of fat acid with long chain alcohol. So in this case we are not talking about glycerol, but we are talking about long chain what, alcohol. And then they are used as waterproofing material by plants and animals. And in plant they form an additional layer on the cuticle of the epidermis of the leaves, fruits, and also seeds. So if you look at the way the leaves are more like shiny. Even the outer part of the fruits, it is shiny, and also the seeds, it's more like shiny. So all that, it's because of the presence of this wax, and this wax is just the ester of fat acid with a long chain alcohol. And then if invertebrate animals, they are found in the skin, the fur, the feather, while in insect they are part of the exoskeleton, which is made of chitin. I think chitin was also explained when we were talking about carbohydrates. So I think you people are familiar with what chitin is. Okay, so this is how wax is formed. So this is a fat acid, and this fat acid will react with a long chain alcohol, and then they will react to produce that particular wax. So beeswax is also a special type of wax which is found in honeycombs, and the bees use it to store honey and to protect the larva and the pupa. It is used as a grazing agent for fruits, a coating agent in cheese and chewing gums, in lip balm and the lipstick. So for our ladies who uses lip balm and the lipstick, this guy is also being used as a coating agent. Okay, the carnauba wax, which is also called the Brazil wax or palm wax, is a wax of the leaves of the palm tree. It is used in automobile waxes, shoe polishes, instrument polishes, floor and furniture waxes and polishes. And then so I think this is direct, and you guys can make some few research on this. Let me quickly explain the last one, which are the steroids. So steroids, like we said, steroids are normally hormones. So these are special lipids compound with 4-ring structure which are found in plants, animals and the fungi. They are special lipids. They are special lipids, and they have the ringy structures, 4-ring structures. So these are just skeletons of steroids. So this is the skeleton of steroids, and then a good example of a steroid, which is what? Cholesterol. You can find cholesterol in the cell membrane of a cell. So the large number of CH group makes the steroid non-polar. So the steroid are non-polar, and the most common type of animal steroid, like I've said, it is the cholesterol, which is the important part of the animal cell membrane structure and the function. So cholesterol is also a precursor to fat soluble vitamins and steroid hormones. And steroid hormone, you can talk about the sex hormones. So females have sex hormones, male also have sex hormones such as testosterone, which is found in the male, and we also have in female such as the follicle stimulating hormones and many other hormones which play a role in issues of sexual activities. So these are just some types you have to consider. I think here I just tried to look at the steroid and also the waxes. So steroid, this is how they look like, which are waxes ester made up of alcohol chain and a fat acid chain. And normally I've already explained that then steroids are characterized by a 4-ring structure. So there is a little bit difference if you compare them, how these guys are looking and how those guys are looking. And when you look at steroid, they normally act as the hormones, which means that they play a role in messaging. So they are more like messengers. An example of a steroid hormone I talked about is cholesterol, and this cholesterol is found in the cell membrane, and it plays a role in membrane fluidity. I think this I explained it when we had a revision class. Okay, so in our next class I won't be explaining the notes. In the time we'll be doing the revision. So I'll focus on exam questions. This is where we are going to look at these guys, like identify the lipid that has a charge component. So the answer here is just the word T, phospholipids. So phospholipids have a charge component. Okay, triglyceride doesn't have, wax doesn't have, cholesterol doesn't have, glycerol doesn't have. Then these are some of the questions we'll look at on lipids. In the meantime, ladies and gentlemen, thank you very much for joining me. For those with questions, you can ask right now, but for those who will have questions after the class, you are free to WhatsApp me on my lines. For now, I end here as I wait for you guys who are in class to ask questions.